So good morning all. On behalf of Amity University Haryana and SPCI, I welcome you all in very important session, COVID-19 and Indian perspective. So in this session, we have very eminent speakers, Dr. Anurag Agrawal, Dr. Sanjay Singh, Dr. Varadrajan and Dr. Lalit Kishore. And this session is chaired by Dr. Sudhanshu Brati. So Dr. Brati is one of the eminent scientists and great academician of the country. Dr. Brati is Director of Regional Center of Biotechnology, and he has an enormous contribution for science area of immunology and virology. Dr. Brati is known for his study in field of RNA virus replication and vaccine development. Dr. Brati is an elected fellow of National Academy of Science, Indian Academy of Science, and elected member of Guha Research Committee and recipient of various awards. So on this word, I welcome Professor Brati, and this chair is ready to you. Over to you, sir.
Thank you very much. I hope I'm audible, right?
Yes,
yes,
yeah.
you are.
Okay, good morning, everyone. It is my proud privilege to be chairing this session, which has four very eminent speakers. We have Dr. Anurag Agrawal, Dr. Sanjay Singh, Professor Raghavan Varadrajan, and Dr. Lalit Kishore. These four speakers speaking broadly on the contemporary theme of COVID-19. We have the first speaker, Dr. Anurag Agrawal. Let me introduce him. Dr. Agrawal is graduated in medicine from the All India Institute of Medical Sciences, and then he has done PhD in physiology. He has been a faculty member at Bangalore College of Medicine. He joined IGIB in 2007. His research interest focus in the area of respiratory health. He has extensively used information technology in health, especially specifically the interface between digital medicine, machine learning, and public health. And as we all know that he's in the forefront of COVID-19 war on behalf of India and the scientists of India. And it is only apt that SBCI has chosen him to speak about COVID-19, specifically the genome surveillance. So Dr. Agrawal will be talking about COVID-19 genome surveillance, and I invite him to present his talk. Over to you.
Thank you very much, Danshu. My slide should be on screen by now. Yeah. It's a great pleasure to address this very distinguished gathering on a topic that has become increasingly close to my heart over the last 1.5 years, as genome surveillance. I added these extra words, lessons from Delta and Omicron. And before we started, Professor Ganguly was pointing out that genome surveillance alone is not just about sequencing, it's about much more than that. And I believe that's exactly what my talk will show today. So let's get started with this picture. Young women, girls wearing masks. It's not from today. It's from more than 100 years ago. It's from the time of the Spanish flu running through the world. And interestingly, the Spanish flu never actually started in Spain, just happens to be a name. And you see these headlines from newspapers: war on influenza, keep church windows open, draft men to be first inoculated for the flu. So vaccines, ventilation, masks, sounds just like, the more things change, the more they remain the same. And why wouldn't they? We are, after all, speaking about one more rapidly transmissible respiratory virus. This was the very weird thing many years ago with the Spanish flu. You can see that very young children and young adults were the ones most likely to die. The risk was actually much smaller for older people after considering all other possible causes of death. And that was thought to be because these people who were old had seen enough similar types of flu viruses over the course of their life to have effective immune responses. The very young had not. And this weird spike in the middle was because there was a slightly unrelated, poorly similar flu that had occurred in the lifetime of these people, which gave them high immune response, but not the ability to neutralize the virus effectively. All of this will become important as you go through succeeding talks in this session, but I thought I would point this out. Not only must you have immunity, you must have the right immunity. Now, the SARS-CoV-2 pandemic has been a bit different. You can see it's pretty much completely age-related increase in morbidity and mortality. It talks about how before this particular pandemic, very few of us had any relevant immunity at all. And COVID-19 is much milder than the Spanish flu at a per individual level, but huge numbers are getting infected because of a respiratory virus with minimal prior immunity. And the evolution of the virus to fitter variants, and that's where genome sequencing comes in, with greater transmissibility, reduced susceptibility to immune response induced by previous variants, whether natural infections, whether vaccines, is a story that we have been seeing again and again over the last 1.5 years. And these massive surges by fitter variants has been overwhelming healthcare systems, despite so far no real change in a low per person infection fatality rate. So why does genome surveillance suddenly become important? If the virus is mutating, changing, becoming fitter, escaping immunity, you need to know these lineages, you need to know the new mutations, you need to find their variants of concern, you need to investigate them if they're not yet of concern, you need to predict changes in infectivity, severity, immune escape, and then of course, beyond predicting from the sequence, you need to measure it. And the last, which is relevant to the One Health concept, you should be tracking jumps between animals and humans in both directions. Otherwise, this idea of bringing these things under control is never going to happen. And this concept is far beyond just SARS-CoV-2. It's a new paradigm for the future for all pandemics. And the WHO has brought out important documents and interim guidances, the most important of which is the following. A genome surveillance program does not stand in isolation. Only if you have sufficient capacity in diagnostics will you generate enough number of cases. Then you need to have ability to sequence those samples. No samples, no sequencing. And after you generate the sequence, you need to be able to analyze those sequences properly and preferably along with other important clinical metadata. Otherwise, you'll never find out what is really happening until it is too late. So SARS-CoV-2 genome surveillance efforts in India. 2020 saw individual efforts by multiple labs. We perhaps did not emphasize the importance of sequencing early enough. But anyway, later on we caught up and 8,000 samples roughly were sequenced in 2020. A new variant called B.6 was identified from India. It was not a variant of concern. If anything, it had slightly reduced fitness. 2021, with the news coming around the world towards the end of 2020 about variants of concern alpha, beta, gamma, led to everybody coming together into a national sequencing consortium in SACOG. Professor Vrati is on the advisory committee. And over this period, over 80,000 samples were sequenced, analyzed, made available on the portal, which I will show you. Kappa and Delta, novel variants of concern were identified in India. Biological studies were completed. Information was shared for global response. Could have been faster, yes, but at least it was done. So I'm going to spend 2020 in just one slide. 2020 saw one particular mutation called D614G, called G strain on next strain, and basically called B.1 on Pango, which we use much more commonly now, which took over the world after April 2020. If you really want to think about VOC, this was your first VOC, but it never got a name because, by the time sequencing started happening around the world, it had already taken over. And it did not look very different anyway in terms of clinical things from the previous one. But actually, when you look back, the ACE2 receptor binding, the ability to spread was far greater for all lineages afterwards compared to the ancestral variant that came out of Wuhan. So this was one big event that occurred without us even knowing it, and no actual name has been given to this. The story of sequencing and VOCs from then to now is from alpha to omicron. D614G is the background for all current VOCs. And I'll show you a better picture of this later. But fundamentally, what you can see is that alpha, beta, gamma are a bit similar to each other. Delta came in a completely different direction and with a greater distance, but look how far Omicron is from all of them. So, because of cross immunity, these variants, alpha, beta, gamma, gave decent cross neutralization to each other. Beta gave very good cross neutralization. Delta was a bit different. So everybody who had gone through, big outbreaks of any of these three still underwent outbreaks of Delta because the sequence changed quite a bit. And Omicron is so different with so many mutations that I will show later on that it has been able to break immunity induced by all of the previous variants. So let's talk about an actual experience of sequencing and the course of pandemic in India 2020-2021. So this is from a website that is now almost closed but taken over recently by IIT Hyderabad. So what you see is, let's look at Delhi. Delhi had a first wave and two minor waves, and then what we would call the second wave of India from the left was actually Delhi's fourth wave. Before this started, in early 2021, the city had reached pretty high seropositivity. 42% in the CSIR cohort, 56.13% in a much better district-wise stratified sero survey of Delhi. It ranged from 50% to 62% across 11 districts. Vaccination had been started, and there was a general sense that if an outbreak occurs, it may not be that severe, because we know that people who are previously infected usually have mild reinfections, and a large amount of the population had been infected. Such an optimistic approach was contrary to what had already been seen in Manaus, Brazil, where despite about 50 to 60% seropositivity, the rise of gamma led to a very, very severe wave. And the reason was that gamma had many mutations that would allow it to escape the antibodies raised to the previous ones. So around this time, little bit around, let's say, February, March, a new variant was being seen in Maharashtra. It had these two particular mutations I've highlighted, L452R, E484Q. 484 site was mutated in beta with a very severe immune escape. 452 was mutated in a previous version in California, had led to big outbreaks. The combination was expected to be more severe. And indeed, by March, this kind of data had become available, that the rise of what was this lineage was called B.1.617. The rise of this particular variant was correlated with the rise in daily positive cases. Was it conclusive? Probably not. But this is roughly what we knew in March, that there is a correlation between the two. At that point, it did not look as bad, and that is understandable because what came out in Maharashtra was actually not Delta, was Kappa. So based on the inputs provided to the viral evolution group, B.1.617 was recognized as concerning. It had two split lineages, 1.1, later called Kappa, VOI, and 1.617.2, later called Delta, which lost the E484Q but gained T478 in spike. And this T478K, along with another mutation you call P681R, were absolutely critical towards some of the properties of Delta. So fundamentally, these mutations at the level of sequence analysis, and this is work by Lipi Tukral at IGIB, looked concerning because they had immune escape. So when you try to in silico bind antibodies to them, they were not binding well. They had stabilized receptor interaction with ACE2 receptor. And importantly, they had accelerated cleavage of the S1, S2 spike region, which is important for viral entry as well as for cell-cell fusion. So the mutations looked concerning. Clinical data, of course, by the time it was fully generated, it was possibly a bit on the late side. But here is something again that makes you understand how even small clinical data can make you understand earlier without waiting for a huge amount of data that shit is about to hit the fan. This is a group of doctors, mostly fully vaccinated. So you can see dark blue is fully vaccinated, light blue is 1, and very light is 0. And amongst these people, there was a big breakthrough cluster formed, where out of 35 doctors, 25 got infected by sitting and having a meal together or coffee together. Now, this made us realize in April that something was happening and vaccines would break with this particular new variant. All the illnesses were mild. So it was not that much of a problem. The problem was the idea that 50-60% immunity will take you to herd immunity was now gone. This virus would go through All these people who were immunized thought to perhaps serve like a wall to the virus and reach the unvaccinated high-risk people. And India only had 5 to 6% vaccination at that point in time. And even with 50 to 60% seropositivity, that still left you with a very large number, even in Delhi, of people who were vulnerable. And all of you know what happened after that. This paper was later published. I'll talk a bit more on that later. The COVID surge in Delhi looked something like this. You can see test positivity rate went up to 30%. These types of numbers you're currently seeing with Omicron. You can see that hospitalizations and ICU admissions went up in sync. Look at this arrow. Look at this flattening of ICU admissions. This is unnatural. It basically occurred because there were no ICU beds left, pretty much in the city. And there are multiple releases by the Delhi government talking about how ICU beds became limiting, and this unnatural flatness tells you the number of people, and that's a large number, who needed ICUs but could not get them. That is what an overwhelming surge of infection does. And that is why it is important to continuously find, analyze, and describe variants of concern. The actual death rate. So you have deaths here on one, the red ones, and the blue is telling you the cumulative cases. The CFR, the case fatality rate, is the ratio. You can see it barely changed from the past. And this was basically the rise of Delta. This paper is also published now in Science a few months ago. And the Delhi outbreak was fundamentally caused by Delta. And all of you know this, I will not spend much time on this. Delta then rose across India, driving the second wave all over the country. Those parts that prepared better did better. Those places that could not prepare or did not prepare did worse. Some parts prepared so well that their seropositivity was very low, and they kept on having cases until much, much later. And you know which states I'm talking about. So INSACOG portal shows a lot of the data that is available with us. Currently, it stands around 82,000 sequenced analyzed genomes. The large chunk of it comes from Maharashtra, which has an independent state program that sequences almost 3,000 sequences per month. It is important, I believe, for numbers to be in this range if you really want to find a variant at a low enough frequency. The less you sequence, the higher the frequency of the variant needs to be before you find it. And if the variant is truly very bad, by the time you find it at 5% and it's doubling every few days, you have no time left to react. And this is just the molecular biology for those of you who are interested. The CT values are much lower with Delta, corresponding to about 10 to 50 times higher viral load. The same thing was seen in cell cultures in our colleagues' lab at Imperial and Cambridge. There was much greater infectivity of Delta over Kappa, which is why Maharashtra did not actually do as badly. But then the Delhi wave was a much more severe one. And because the two are very similar, you would expect that previous exposure to Kappa protected you from Delta. So that's another thing that is luck. Vaccines were greatly reduced in effectiveness. Again, we know this. This is insight from vaccinated healthcare worker cohort that we have. This paper should be coming out in Nature Communications soon. We found 25% breakthrough rate despite good antibody titers after 2 doses of Covishield. The breakthrough rate was a bit higher even than this for Covaxin. That paper has just gone on medRxiv yesterday, so you will see that. Most things were asymptomatic. That is the beauty of vaccines. So vaccines prevented severe disease effectively, but were not effective at blocking transmissions as we expected from the sequence and the in silico work. The T-cell immunity that is very important for reducing disease severity remains relatively intact, even when antibodies don't bind. And I will not walk you through this figure, it's a bit complex for that. But most healthcare workers, even without knowing it, who only had been immunized by Covishield, which only gives you antibodies to spike, had developed antibodies to nucleocapsid and had developed good neutralization, actual culture neutralization to Delta without even knowing it. That's the part that gives me a little bit of hope as we go forward towards Omicron. We have so many vaccinated and infected people, whether infected followed by vaccination or vaccination followed by infection, that they have very, very high antibody levels. And even though I again think that we will significantly lose neutralization, it will still be okay for a chunk of people, but not all people. I must emphasize that. And this is a very interesting analysis that was done along with Imperial that showed that there was both increased transmissibility by a factor about 1.7-fold compared to Delta. compared to alpha, which anyway had 1.5 times greater than B.1. And immune escape also was increased to the level of about 25%. When does it end? This is data from the CSR cohort showing that in our cohort, unvaccinated people reached 88% seropositivity by the end of the second wave. Many of them had reinfections. Our estimate is around 28%, out of which 10% is RT-PCR proven. That's how frequent reinfection was. I know that you get various numbers from a variety of agencies talking about reinfections being only 1 to 2%, but this too is already published in the same science paper I was referring to. True reinfection rates, even with Delta, were much higher than what is known. And it is obvious. If you only find, see, India's infection rate is 2.5%, 25,000 per million based on known cases. It is 66% based on serology. So about the true number of cases is 20 to 30 times higher than the known number of cases. So obviously reinfections are also grossly underestimated in previous times as well. So I would suggest to you that you rely upon this data, not the other. And it's a global problem. It spread rapidly across the world. Vaccination saved lives everywhere. The mirror image is vaccination and death. So what you see is in the beginning, the reflection was big. So blue are infections, red is death. And after vaccines, the red reflection becomes much, much smaller, except in countries which are poorly vaccinated, where the death reflection continues to be high. And you do lose efficacy even for severe disease, but not as much as you do for infection. Let that also be very clear. There is nothing called no loss of efficacy against severe disease. For example, if you have seen the recent paper from ICMR, a very nice paper done along with AIMS, in which they show that for severe disease, efficacy was 80% for Covishield, 69% for Covaxin, and it used to be nearly 90% against the ancestral variant. So while they will remain highly effective, even against variants for severe disease, it is not going to be 100%. So we'll just go past that. And basically, this is a nice graph again made by Lipi Thukral. I should have put the credit over here. The virus has evolved, but I want you to take a look at Omicron. The N-terminal domain is important for some antibodies. RBD is critical for antibodies. S1, S2 area is important for cleavage. And what you see is that Delta had already given you a lot of mutations, but all this is nothing compared to Omicron. Absolutely nothing. Omicron will escape antibodies. This entire areas, NTD, RBD, are so heavily mutated. It will have decent cleavage, although it may not be perfect because of the mutations. And already there's a paper out by Ravi Gupta's lab yesterday. I have already posted on Twitter. Please take a look there. But this is how South Africa found Omicron so fast. South Africa generates the sequences for the variant in the same month the sample is collected. A sample was collected in the province called Gauteng on November 9 or November 12, depending on different sources. It was sequenced by November 21. By the last week of November, we held a meeting at WHO, and it was very clear that a rise in cases in Gauteng was being driven by this particular variant, and the rise was looking like this. And, reinfections were starting to rise. They had maintained cohorts of people where they could calculate reinfections very fast within November itself. So you can see, this is a meta-archive paper that was put out, and you can see that they could see that reinfections were starting to rise. And based on this increasing reinfection risk, evidence was made that, VOC call could be made early. And then, of course, you all know the story. It has gone since then. Omicron has really, really shot up. It is now looking like it's coming down. Preliminary data from South Africa did suggest that the share of COVID patients requiring severe care is lower than in past waves. But I would like to emphasize again, it is not zero. It is lower than in the past, but it is not zero. It is too early to become too happy about Omicron. I'm going to end now by saying that sequencing got us some information about Kappa and Delta. It took time. It was still a young program. Sequencing in a more established program in South Africa found us beta, helped in alpha, and found Omicron within the same month that Omicron first sample was seen. And even when you look at the molecular clock of Omicron, it is only in October at latest type of a virus. So within a month, the entire world got informed, and today we are able to plan measures and build better vaccines. In the end, immunity will determine everything, but there will be need for additional tests to find these VOCs quickly and at scale. You can't sequence all samples, but you can use innovative RT-PCR, and there is a very nice paper by Debojyoti Chakravarti. There is one short letter in Lancet and a longer paper in eLife, where CRISPR-based diagnostics can also be used in a completely digital way to find these variants fast in absence of sequencing. With this, I will end. Thank you very much.
Thank you, Anurag. Are we going to take questions now or?
Yes, sir.
Yeah.
Please.
Okay.
2-3 questions quickly.
So thank you, Anurag, for very nice presentation and bringing us up to date on Omicron story. So we can have a couple of questions from the audience. Anybody?
May I?
Yeah, Sunit, please go.
So it was wonderful presentation, Dr. Anurag. If I could see your slide of mutations which were represented in the blue color, where you have shown really alpha, beta, and then Omicron maximum mutation. So, yes, this one. So don't you think time is not far away when these mutations itself become suicidal for this virus? Because the way this is happening, I think it will, after some time, it might become just endemic. That's what I want your comments on.
Thank you so much. It's a wonderful question. And in fact, there's a very nice pre-data released by Ravindra Gupta's lab, our collaborator in that Nature paper. What they're showing with pseudovirus is that while this particular spike, which is heavily mutated in Omicron, has gained the ability to escape antibodies, its ability for S1, S2 cleavage coming over here has become less efficient, which means its actual entry might require a greater receptor density compared to earlier, and its cell-cell fusion capacity may also decline. I have just put this on my Twitter earlier today, so please take a look at that. It's exactly what you are saying. So what it means is that it will require a higher receptor density to enter efficiently, which would fit the Hong Kong data, that it is proliferating better in bronchial epithelial cells compared to alveolar epithelial cells. If so, it will become more like a URI and less like a pneumonia, which also fits this other data I was showing you, where severity seems to be lower. But I would put a caveat to everything I'm saying. Maybe is not is. And given how many people it can infect very, very fast, even a smaller percentage of a larger denominator, we never know which direction it's going to work out. So while I do believe what you're saying is correct, eventually the virus is going in that direction. It is possibly too early to be too happy for Omicron, and I would still think vaccines remain the way forward.
Thank you very much. Highly appreciate it.
Hari, you had a question?
Sir, for example, can I go ahead now? Say, Anurag, I think it's a nice presentation, though I feel that even scientists will get scary, those who know the virology even. But, my question is something, what happens to the human influenza virus A and B? What is the genomics answer to that? I mean, what do we say that?
So influenza is quite different.
Let me add another question. But when you did all these studies on genomics, did you also not bother to take some 10,000 or 1,000 sample of normal influenza virus, see that how is that mutating? That's what I want your perspective. So what exactly you think of it?
Okay, so that's a very good question. So GISAID was actually formed for influenza viral evolution. And what you see is the way that the influenza evolves and the way SARS-CoV-2 evolves is very different. Influenza is particularly a recombination-driven flu, basically viral evolution system. there are some hints that even SARS-CoV-2 may show recombination, but if so, they're probably going to occur in the same person who is chronically infected, where multiple variants are emerging because of serial mutations, affording the chance for a low probability recombination over time becoming a high probability. So the two viruses are very, very different in this. And in fact, even the genomic analysis should ideally be different. And something that, Sudhanshu and I talk about often is why, in this finding deletion that are hidden below primers based on amplicon-based sequencing is more important than that. But that's going to get very technical for other people. So we are looking at it, but, we are primarily used to be a human sequencing institute, and we haven't done that much influenza.
Thank you.
I think one last question we can take.
Hello.
Yeah, go ahead.
Yeah, so suppose Omicron in spike protein, 30 mutations reported. But as compared to Delta strands, so few mutations I think is effective for the increase the infectivity as compared to Delta strand. Okay, like 2 mutations as modeled by the all 30 mutations as per Professor Balram Bhargava. Sorry, Professor Balram from ISC Bangalore, he reported. So my question, whether this Omicron, how about the vaccine as effectiveness, COVID shield versus or co-vaccines, means the better community is how much protected, means against the.
So you ask a tough question, but yes, the higher transmissibility mutation that is new is possibly 679 for S1S2. And of course, there is the N501, but it lacks the L452. So nobody exactly knows in true transmissibility, is it worse than Delta? Might be 1.3 times is the best estimate. The real gain comes from immune escape, not really from real transmissibility. Immune escape is really driving its advantage against Delta right now. And if you know that immune escape is driving its effect greater spread than Delta, you know that vaccines will lose efficacy very fast. Unfortunately, we don't have the data. UK has the best vaccine data so far because the first highly vaccinated country in which the virus has spread. AstraZeneca 2 doses were nearly ineffective. In general, AstraZeneca has done better than inactivated vaccines in every study so far everywhere in the world, including in our own data from ICMR and AIMS. So I would expect, without knowing, that Covaxin will have the same problem of loss of preventing infection. I would say again that ability to prevent severe disease is different from the ability to prevent infection. And we should assume it will be high. Will it be 100%? No. Will it decline? Yes. But it will still be high, I think. That's what the data shows so far. So, and regarding vaccines, better people than me will come to speak next.
So one last question. How about the booster dose? Can you comment something about the booster dose?
That's a controversial one.
Raising immunity is important to prevent infections. The gain will be high if it is plausible. The gain for preventing severe disease will be small. The priority remains vaccinating the unvaccinated and finishing the second dose for those who have got one. But high-risk groups like older people and healthcare workers and immunocompromised, the debate is ongoing. But it's easy for me to say yes. The people who are in the committee will have to not only say yes, they'll have to say which vaccine, when, by which mechanism, by which platform. So those nitty-gritties are taking time.
Thank you, sir. So much nice presentations and updated information. Thank you so much.
My pleasure.
Sudhanshu, if you have time, Professor Ganguly wanted to ask a question.
He promised he will not speak.
No, he was raising his hand.
I just want to give one. I want to give just one information that today the Imperial College has come out with the data that Omicron causes severe illness. So as Anurag said, that don't be complacent. So you see different things. One more comment I want to make, that even in a very highly vaccinated community, you are seeing Omicron and Delta is coming with another wave. What is worrying is, as Anurag said, very few people said that Actually, Tony Fauci said that if we get the herd immunity has to be around 85% that came out through the modeling of the measles infection. And only then it could be perhaps of any significant part of this. But here, what we have seen that, and I'm worried about myself. I'm now 80 years old and my antibodies have fallen. I was one of the first to be vaccinated. It has fallen to a level which is very, very uncomfortable. So the logistics permitting unless we take booster, as now some countries have realized where there is a surge now, who have been vaccinated quite heavily but are having another surge. This is because the immune levels have come down. And one more thing we are not talking, and this is my last comment, that there are vaccines and vaccines. Not all vaccines work together. And there are options. One particular thing came up by the CDC, that the 2 messenger RNA vaccines are better than the Johnson & Johnson vaccine. Not only for other, but also because it causes blood clots. But I know about other vaccines which have very severe side effects. 1 in a million or very few people get it. But we need to ultimately come to a decision that which vaccines are better and where the production need to be boosted. And as there is Anurag said, or I don't know who said this, that it will be possible to make messenger RNA vaccines very quickly by Indian drug companies who have never made a vaccine before. So we need to have a strong thing. But Anurag, I really like your paper. And the GISAID, I was one of the founder member of GISAID. At that time, the two different, and both your institute have both the capabilities. The two different capabilities came up. One was Swiss Institute of Medicine, which has a genomic and bioinformatic skills, and the institute in Basel, which had proteomics and bioinformatics skills. Because as you know that apart from the ACE2, this virus has several other binding points, particularly the beta integrin, which comes between S1 S2 by the furin cleavage. And so to really combine these two skills with the sequencing, perhaps will bring out more answers, and your institute has both these skills. But I will compliment you. I really like your presentation. So I take back my earlier comment that sequencing doesn't do much. I eat my own words and
No, sir.
apologize.
Thank you very much.
Thank you.
Thank you, sir.
Thank you very much. I think we will now conclude this talk. Thank you, Anurag, and move on to the next speaker, who is Dr. Sanjay Singh. Sanjay Singh is Chief Executive Officer of Geneva Biopharma. Dr. Singh received his PhD in biochemistry from CDRI Lucknow, and after that he worked for a number of years at NIH. He returned to India subsequently and has been at Genova since 2006. At Genova, his scientific expertise has led to several innovations to treat life-threatening diseases. And currently, Genova and Dr. Sanjay Singh have been at the forefront for developing India's first mRNA vaccine against COVID-19. And we're going to hear on it from Sanjay, although there was some news about this vaccine in the newspaper this morning. So maybe he has already leaked some information, but let's hear from the horse's mouth. Sanjay Singh, please.
I'm still a scientist and believe in the data. So you might have seen very less into press. I'm not still a businessman. I have been asked by Professor Rajan Prasad in the morning. Otherwise, I was wearing a shirt. But it's all right. Go ahead. So thank you, Professor Prasad, for inviting me. And what a pleasure to meet all the peers in the field here. So in this program, when the organizer asked me to I speak on this COVID-19 and Indian perspective. So I'm going to talk a bit about the mRNA vaccine. I think this is a few of the rare forums where I have gone and talked. And for me, it is a very proud moment because I have attended many SPCs when I was a PhD student. So starting from it, so this is the first one. My task has been made very easy by Dr. Anurag Agarwal. So Omicron is a possible worrisome point. We do not know which direction it will go. But if the infections are going on, then let's see where it ends. And it is important for us to prepare. And this is, I'm taking mRNA side, that why mRNA technology is important. I'm more going to talk about the technology part, not on the disease, or just because this is for the young student. And looks like whatever latest is seeing, some relief may come from mRNA, one of the mRNA vaccine, which the latest data has come. So now I'm going to talk about the vaccine platform and how the mRNA get integrated into it. So basically, these are the 4 independent vaccine platforms. First started by Professor Jenner very early, maybe a decade, a century before, where you can take a whole organism, weaken it or kill it, and make a vaccine. So from this, polio, hepatitis, rabies, influenza, yellow fever, many vaccines came, and still a lot of vaccines are available in the market from this technology. And our peers, Bharat Biotech, brought the COVID vaccine against it with ICMR. The second tech-based technology is basically recombinant technology, and this added the advantage because you don't have to deal with the very infectious virus. But here you can go on the gene side and you do the sequencing, and from that you deduce your amino acid sequence and make a protein, make a recombinant protein, make a cell bank, and then start fermenting and do it. So a lot of vaccines are available, and hepatitis B was one of the major things which came out. Now influenza vaccine, many vaccines are there. Then a third vaccine platform emerged maybe 20-25 years ago. But the first approval, rare approval, came as emergency authorization for Ebola vaccine, and then now the second is AstraZeneca or Serum Institute vaccine. So here it is adenoviral-based non-replicating DNA or plasmid DNA vaccine, which is also now Zydus vaccine approved. So each platform has some advantage, some disadvantage, and the way the technology is developing. So now we can talk about the latest platform, which is the mRNA-based vaccine platform. So here, basically, there are certain unique advantage came into it because you ask the human body itself to make a protein which mimics the most native structure of the viral protein. So hopefully, the thought process was that it will give, if you deliver it well, the thought process was it will give the best functional immune response. And this was the premise for this whole mRNA. Now, what are the advantages and what are the difficulties into each platform, and why this field is moving into it? So if you see on the bottom, one thing is time to manufacture, and second is economics of manufacturing. So when we take the first technology, which is wholesale, so you have to deal with a very highly infectious virus. You have to propagate it, inactivate it, then you have to estimate the potency, all these things. And so this is a technology, but it still is used, but this requires huge facilities and huge curtailment BSL-3 or BSL-4 facilities. In case of vaccine 2, which is the recombinant one, yes, you make the MCV, master cell bank, all that, and then it is a proper biotechnology, huge operations. Then when you come for vaccine 3 technology, which is basically non-replicating adenovirus or DNA platform, so you have to produce huge quantity of plasmid. But in template 4, if you see that vaccine 4, here you need a very small quantity of DNA, and then it becomes in vitro transcription, and you can do in a tube, or you can do in a big bucket, or you can do in a big vessel, and then very few quick steps for mRNA. The problem with this was that how the mRNA has to be delivered, that complexity was solved, and somehow COVID gave opportunity for this vaccine to fare. And what I told you, that if it produced the most native structure, which is most important for the vaccine, then this vaccine, this technology has proven the safest till now and the most efficacious. So now mRNA vaccine platform. So you can see that though the most safest and most efficacious vaccine technologies is available, vaccines are available, but it was very limited distribution all over the world. And if you see if this has to go all around, the vaccine coverage is very limited, and looks like by 2023, what Dr. Anurag said and what Professor Ganguly said, if you need to have 80% herd immunity, then reaching the vaccine is very, very critical, and which doesn't look very soon in time to come. The reason behind it, when we talk about it, there is nobody said one particular thing. We all will be safe when we all will be vaccinated. Even one zone of the world is non-vaccinated, have non-vaccine, which is not of super efficacy, the virus will keep it multiplying. This virus has a unique problem. It is jumping between human and animal, and continuously this jump, it is modifying much faster as compared to any other viruses we have seen in the past. So the same thing what Dr. Soumya Swaminathan said, that it could have been stopped, but why what is the problem into the mRNA technology and why it is not reaching to this? So I'm introducing our own program. This program we started very early. I think I wrote to Deputy Secretary somewhere in Feb, and our program was reviewed, and finally part funding, seed funding we got from DBT. And at that time, there are certain presumptions we put together, and at that time we were not knowing this vaccine will be successful or not successful DNA platform. But we changed two things. One, it was we used self-amplifying platform, and which is different from existing vaccine. And second, we didn't encapsulate our mRNA into the nanolipid. We put it on the surface of it. And there was a reason, because we wanted this vaccine can only reach to the global reach if we have stability of this vaccine and to it. So this was one of the huge thought process behind it. And then it can be used for mass vaccination, because minus 70-degree supply is not available all over the world. It is very limited to certain developed countries. And then also there is a possibility for booster dose, because we were knowing this is going to change, is going to move, because it is jumping between human and animal. So for this, give advantage, because you just change the plasmid and you make the next vaccine. So just for the sake of that, what kind of data made us to go forward? So we produced the cGMP batches, and we did 2 immunization, day 0 and day 29. And if you see on the bottom side, we got a good immune response and very good boosting ability into day 29 we did. Then there's a gold standard assay print 50. We came close to 1,000 print 50 titers, which is very, very good, because our normal conventional sera gives around 130, 140, 160 in the majority of the cases. And there was correlation between antibody titers and the print titers. But running a print assay is so difficult, because we don't have BSL-3 facilities. We have to depend upon some of the outside labs. So there was one surrogate assay also came out, and it was published in Nature Biotechnology at that time. And this is neutralization assay. So this assay, when we tested our sera on day 14, around 75%, then it gone to 80%, something 90-95%, because you cannot get 100% inhibition into it. But when we compared the neutralization titer of the convalescent people and with 2 new people, we found that somebody who came back from the ventilator, they had the highest, around 60% inhibition in this assay. And then another convalescent was having 50%, another was having 40%. Somebody who got twice infected, that sera was also having around 60% of the inhibition. So this gave us the confidence that everything is working, and we should move forward with this. Another most important part is not only humoral response, as Dr. Agarwal also said, the cellular response is very important. And because of time, I just want to focus yourself only on the G part. And here we were able to demonstrate that spike-specific germ central B cell response we are getting. This was one of the few data which demonstrated it. And that gave a confidence that yes, our technology, which is basically the self-amplifying, because we are giving a very low dose and slowly the response comes. So we are able to hit the spike-fixed germ center of the B cells. And that was very encouraging for us. So from here forward, we gone into human clinical trial. We completed phase 1 trial, then we completed phase 2 trial. At present, we are running through a phase 3 trial, which is 4,000 volunteers. We have already completed the first dose as of today, as we say, and looks very safe. From phase 1 to phase 2, immunogenicity looks like great. And phase 2, we have done a comparative. The one arm was our vaccine, mRNA-based, another arm was Covishield. And let's see, we are going to do unblinding through DSMB next week. And once unblinding comes, then we are going to present to DCGI. So here we are, but I will go into more deep into it because that's what this technology offers. So the basic problem that why somebody came from New York Times and they wrote first that this technology can never go to emerging markets or less developed countries. And that was very challenging for a person like me, because as I said, I'm still a serious scientist. So first problem was ultra low temperature. The second is ease of manufacturing. Can we create those kind of facilities manufacture? Third is the biggest problem is supply of raw material, because all the raw materials required for this technology need to be imported. Then can we scale it up? And then also because we feel that we can take this technology global, because if we solve these one by one, then opportunity is there to go global. And we are in discussion with Gates Foundation and WHO how we can do this. So first problem, I would like to say this was the very, very important problem, ultra low temperature logistic. So mRNA vaccine require -70, which 2 vaccines on the market, and we thought that can we make it 2 to 8 degrees centigrade. And here I would like to bring the scientific knowledge. So if you see the part B, basically mRNA is put inside the small pockets which are still containing waters. And here in a big ball, you can see the nanolipid inside that mRNA is trapped. And if you see in a more defined manner, this paper was published in the middle of this year, when we were going into this, that these still contains these channels of water. And the biggest problem is why mRNA is unstable as compared to DNA, because it has particular OH bond and it makes it susceptible. So all the information in the body is kept in the form of DNA, but when DNA want to send the information, it's sent through RNA, and RNA convert into the protein. And this is the way the nature control it. And if you see what is the basic science into it, this is particular phosphodiester bond. And when hydroxyl group is there, in presence of water, this bond gets broken and the bases are released. This was this very simple chemistry was there, and where we worked out. And finally, we made the lyophilized version of the vaccine. We removed the water. And now if you can see, our vaccine is stable for 180 days at 2 to 8, and even 25 degrees centigrade, it was stable for 30 days and so. So if we see on the first side, which is the blue side, you can see this was -80 Fujian vaccine. We thaw it and injected. But when you make the lyophilized vaccine, we have done this experiment and we found there is no difference into the immunogenicity, there is no difference into the titers, and even better neutralization titer. So if same assay we have used, surrogate neutralization assay, if you see where we say 40 to 60 is convalescent, we got it, but only thing we found some extra functional immune responses coming faster is coming on day 28. So this is the way we solve it. And then coming to the ease of manufacturing. So here, if you do entrapment into mRNA by pressure, you have to make these nanolipids and you have to throw the mRNA into it. There is a lot of loss. The recovery is around 30-35%. But what we did, we created the CLN release and we put it on the surface. If you see the picture, this is on the surface of the positive and negative charge. The mRNA is kept on the surface. This was very, very challenging for us. But this was basic because by when we do the lyophilization, we can take out the whole water and add when water, it comes back into the same format. So this was one of the challenge which we tried. Second, the challenge we try to solve. And this also gives a near homogeneity. And this, we were struggling when we were making it into 550 microliter, you can do it, 5 ml, you can do it, but we have to make in thousands of liters. So now whatsoever do in 50 microliters, same we can get the similar kind of product at 2000 liters. So this is another problem we said. And as I said, there is no water available, so vaccine is a rock solid step. The third is the supply of the raw material, I would like to say, and this is for young students who are hearing it, or the policymakers which are hearing it. Our self-amplifying platform gave us that we have to go with a very low dose. And why? What is difference into it? So if you see Moderna and Pfizer, this is the base 5 UTR and 3 UTR, and they have used some modified nucleoside. And CureVac use same, but they have not used modified nucleoside, and they use a very small dose. In case of us, we have put a self-amplifying assembly. So same, when we give the construct, it goes for 5 to 10 cycles, it produces a lot of mRNA. Slowly it produces, and that gives some advantage to us. So we have to go with a very low dose. So at present, we have completed, when we completed our phase 1 trial, we realized that 10 microgram dose is very good for us to go forward as compared to BioNTech 30 microgram, and another one is
mobile phone. Now I wish to join through my laptop. So that's why I was.
Sure, sir. I'm gonna mute you and push you live.
There is some disturbances coming.
Please stop talking. Mute it. Devendu, mute the microphone.
So here, if you go the process, we have the simple plasmid, DNA plasmid. We use some restriction enzyme, we use particular 3 enzymes, T7 polymerase, pyrophosphatase, RNase, and then use the DNase to kill the plasmid and purify it and cap it. So basically, this is the process, but all the materials was outside. So we also started a program to do backward integration. And most of the enzymes we have made now in-house are with our people. And this term is scalability was the issue, because we are a small startup company. We tried to do it as much as possible. But today, looks like because of our technology innovation, we need low doses, and also scalability is a simple. So here, a very typical slide I wanted to present. So we have broken this whole unit operation into 4 parts. One is, can we produce enough plasmid so we can store separately? Then a second unit, we can produce through IVT process, the mRNA. Third, somewhere else we can produce our cLNEs, and then we mix together and lyophilize the vaccine. So these are independent 4 operations we can do. And if we have to transfer the technology or take to globally, so at present we are talking to many countries that where we can ship these 2 particular things, and finally mRNA vaccine can be lyophilized sooner or later at those places. So technology transfers, yes, WHO is behind us that we should be part of this, and we are committed to do this technology transfer. So hopefully, if everything goes well, we are going to partner with 4 or 5 regional, one in Latin America, one is CIS countries, one is another part of Southeast Asia, one is in Africa. So let's see how it goes, and we can do this. But at present, we have developed the capacity which will be fully operational by 2022, by which we can produce 1 billion doses a year. So now, one by one, we have solved these things. And in summary, what we want to say, and this is very important, why I said this is going to change the future of vaccinology. These are the left side. You can see cLNE you can make and store, enzyme you can make and store, cofactor, nucleotide, lipid you can make and store as a national stockpile. And as soon as the plasmid variant change, you just resynthesize your gene, put into it, and finally the vaccine is ready. So roughly, if you go around it, if we have to make a new variant, it will not take more than 50 to 60 days before you can inject, subjected to regulatory approval. Now you'll ask why Genova has taken so much of time, because this was the first time we are introducing this technology to our DBT people at RCGM or at DCGI office. Yes, regulator has got up the questions, but the way the data has come, the safety has been demonstrated, emergency demonstrated, hopefully things will go forward. And if God is gracious and regulatory approvals comes, we should be in position to put Omicron within 60 days into the arm, and we are working on that. So now there is another problem which we are thinking and moving forward. So tuberculosis is one of the area, dengue is one of the area, chikungunya is, Hep C, and Johnstone malaria, and these are all with different collaborations. We are taking this platform forward. So I hope the modern technology of mRNA is going to save a lot of lives. Deployable, we have seen in the past, simple rotavirus has made huge advantage in terms of saving the children. So I feel that this is going to contribute significantly to our society. Thank you very much.
Thank you, Dr. Sanjay Singh, for a very nice presentation. Actually, this would be a proud moment for the country when this vaccine gets commercialized, because this is based on a very new technology. And also, this might be the first mRNA vaccine which is stable at 2 to 8 degrees centigrade, making it possible to take it to resource limited settings. So thank you very much for your talk. We might be able to take a couple of questions. So
Sanjay ji, congratulations for good work, and I'm very happy to see such development of mRNA vaccine. Although you mentioned that if mutation is going on for as Omicron, so how about the effectiveness of new strains of these vaccines?
Yeah, so certainly as the data is showing, Omicron can escape the vaccine because our base is D614G. But because there are around 39 mutations out there. So certainly this is going to, this vaccine is going to give a general center response, just like what you see in the native infection when you see. But certainly Omicron will need a different version, because as soon as it came, everybody starts saying in India and outside also that, oh, our vaccine will work, our vaccine will work, because I think they don't deep down into the science. Nothing is going to work. Finally, the BioNTech has also realized, Moderna already said that they are going to make the Omicron version. We also said to the government, we are making on this sequence. We have, this is advantage of this technology. If the new one comes, we have to boost it till we reach to a very high level of herd immunity. Because it is not the how severe infection it is, because X population is having comorbidity. And in case of comorbidity, those infections in these people can become into severe cases. So I don't want to go into detail because this is a great scientific forum. We have seen such a beautiful presentation from Dr. Agarwal. We do not know what Omicron is going to do, but certainly we need to prepare ourselves for preventable. And this is the whole concept of vaccinology. Because once first our 2 vaccines were launched, the office of the government said, oh, we have started the end of the COVID. And you know what after it happens. So we have to, as a policymaker, as a scientist, we should not go and make these kind of statements. We have to be ready, and we should have preventable measures, because our hospital can handle very limited number. If a big number comes into the hospital only for mild infection, everybody will rush to the hospital. Hospital will be choked, and last time we have seen it. So I think government is very cautious this time. I think our policymakers are also very cautious this time. We do not know how it is going to come, because Omicron, which has come already, we see, I think Dr. Agarwal must be knowing, 3 lineages are already coming into from that. What will go with the multiple transmission, where it ends, we do not know. Because Wuhan 1 was not that dangerous. The second D614G was a little bit more dangerous. Because whatsoever taken all over the world, these are the one which has taken all over the world. Delta was taken all over the world. It created a problem. So I do not know how it will come, but we have to be cautious, and we have to be, as a scientist, we can say, can we make some preparation for preventableness? That's all.
Thank you. Thank you, sir. Thank you so much.
is there any other question?
Dr. Sanjay Lalit here from, I mean, it's great to see an mRNA vaccine come out, but my specific question / more of a request is to consider options for indigenization of ingredients as you go forward. It's great to see that there is already a tremendous effort in backward integration, especially you know that mRNA vaccines, specifically both chemical and enzymatic capping technologies, have been kind of held very closely by IP globally. If that can be freed up, I think you would be creating an ancillary set of people who will create ingredients for you and multiple other companies and multiple other vaccines from your own portfolio. I would hope that you are considering that as you go.
Yes, this is very important, and that is because we can make few million doses any day, but when the doses goes to $1 billion, then the situation comes, and our competitors has already taken over all the raw materials is available in the world. So my parent company, which is MCure, we have already made our DOTAP and we have already made our lipids available. So we are, and then we have signed a big agreement with the suppliers. So we are having multiple ways. T7 polymerase we made a couple of months back itself. T7 polymerase, guanine transferase, we are also trying to make our own. So certainly we would like to have total indigenous availability, because if we depend upon even bags to outside, we will have a problem. So this is the whole aim of our effort, and why we are late, because we want to develop a vaccine which will be globally respectful, because otherwise what the West said, oh, you can do this, you cannot do this. No, we are the same people. This science has been not innovated at Saturn. Only thing, if we put the right effort, government's regulator, government, our RCGA members look from a more proactive approach, we can do these things. This is very easy to do these things. You see the Dr. Anurag presentation, this is world-class data. So why not we can make world-class product? So I'm very keen, and our 50% effort are on the backward integration with us, with our partners, and with different vendors in India itself.
Thank you, Sanjay. I think in the interest of time, we will have to not take any more questions, and we'll go to the next talk. Thank you, Sanjay. The next speaker is Professor Raghavan Varadarajan. He is a well-known Indian biophysicist who works at Indian Institute of Science. His field of interest is studying protein structure and folding, and he has contributed significantly to developing vaccine candidates against influenza and HIV. Dr. Varadarajan has several awards to his credit, and he is also a J.C. Bose National Fellow of SERB. Dr. Varadarajan.
Thanks, Sudhanshu, and thank you to Professor Rajendra Prasad for the invitation to talk here. So I'll talk a little bit about our own work on immunogen design for COVID-19 vaccines. So basically in my lab, we work in the general area of protein stability, folding, and immunogen design. And over the years, we have developed ways for stabilizing proteins and also making immunogens for flu, HIV, and most recently for COVID-19. So I'll just give a brief background to two technologies that we use extensively. One is saturation mutagenesis, also called deep mutational scanning, in which we can make libraries where we mutate every single residue in a protein sequence and then couple it to some sort of protein display or phenotypic assays. And this allows us then to find the effect of each point mutation at each site in the protein upon a given property of interest. Now, this methodology is very useful when coupled to binding assays or phenotypic assays. Our interest has been how to use it to improve stability of proteins. And for that, there's no easy direct readout which correlates with protein stability. So we use yeast surface display, and in yeast surface display, the protein of interest is fused to another protein called AGA2, and that is attached on the yeast surface. You can detect the amount of expression of the protein by an epitope tag. Then you can add a binding partner which has another epitope tag, and then you can detect how much of the binding partner is bound, and that's a measure of the amount of active protein on the yeast cell surface. And you can do this with libraries and therefore sort for proteins which have better binding affinity to a binding partner of interest. But again, there's no trivial way to convert this into a stability assay. Okay, so with that very brief background on some of the background technologies, so everyone here is familiar with SARS-CoV-2 and the spike protein, which is the primary target of neutralizing antibodies. And as Anurag has already mentioned, a significant fraction of neutralizing antibodies bind to the receptor binding site. So they prevent binding to the ACE2 receptor. And then following the proteolytic activation, what happens is that there are conformational changes which lead to fusion of viral and host cell membranes and viral entry. So most vaccines are focused on eliciting protective and preferably neutralizing antibodies that block this interaction either directly or indirectly. So now there are there are several different classes of vaccines which are in clinical use. They are mRNA vaccines, non-replicating viral vectors. The Chadox vaccine is an example of this. Once Genova is in use, there will be an additional mRNA vaccine. There are a variety of inactivated viral vaccines and recombinant protein vaccines. And this is our current status in India. Right? We have close to 40% of adults are fully vaccinated, which is, of course, a remarkable achievement, but there's still a long way to go. And sero surveys estimate a much higher fraction of the population has been exposed to the virus. So now there's a lot of data both from clinical trials and actual use in the field. And so a few salient points I wanted to highlight. First of all, the neutralizing titers. So that's the high neutralizing titer is a measure of the amount of neutralizing antibody elicited. These have been, for the vaccines which have been tested so far in humans, have been similar to those in small animals. They have been relatively low for both the inactivated virus and the adenoviral vectors, and better for the mRNA vaccines, and somewhat higher for the subunit vaccine. And higher neutralizing antibody titers are associated with higher protection against infection. But of course, the real concern is against severe disease, and there the differences are, not so stark. And of course, as everybody has been saying, these recent mutant strains are a big concern, and most certainly the Omicron strain will be able to evade neutralizing antibodies to a great extent generated either by prior infection or vaccination. The good news is that relatively low titers of neutralizing antibodies are sufficient to protect NHPs from lungs pathology. And although all the correlates are not known, certainly neutralizing antibody levels are a very clear correlate of protection. And again, as Dr. Sanjay Singh was saying, all the current formulations require refrigeration, and the mRNA especially require low temperature storage prior to administration. So I've just listed, the mutations that are in the various variants of concern. And of course, the Omicron has a far larger number of mutations, as has already been pointed out. So the beta variant, which originated earlier, there was a roughly 10-fold drop in neutralization titers in convalescent sera relative to the B1 D614G variant. And Omicron and Delta was about 3 to 6-fold drop. And Omicron, very recent data suggests, is about, there's a 10 to 15-fold higher drop in neutralization titers. And as others have emphasized, we don't know enough about the effects on transmissibility, ability to cause severe disease, especially in people previously infected with Delta or vaccinated with current vaccines, or in some combination of both these things. So we, early on in the pandemic, we started working on a vaccine based on the receptor binding domain. And this rose out of the reason we decided to target that region is because we knew from earlier work on flu that the most efficient neutralizing antibodies block receptor binding. And subsequently, that turned out to be true. This is the receptor binding domain in the context of the spike. This is the receptor binding domain colored by the regions where neutralizing antibodies bind, and practically the entire surface of the RBD is a target for neutralizing antibodies. These are regions where, about a year ago, neutralizing antibodies were known not to bind, and now we already have neutralizing antibodies known in many of these other regions also. So The RBD is a relatively small fragment of the spike protein, about 200 amino acids, and it contains 2 glycosylation sites and 4 disulfides. And we previously described glycan-engineered RBD fragments, which were expressed in a number of different expression systems. And what we showed was that the protein was extremely thermotolerant, and when it was lyophilized, we could expose it to boiling point temperatures, and upon cooling down, it still retained its conformation. This was published late in 2020. And we also found that the mammalian cell expressed protein showed much better, elicited much better neutralizing antibodies than the PICA expressed protein, which was fairly which was surprising to us. So next, because it's known that oligomerization of the protein increases its immunogenicity, we made trimeric versions of our monomer design. We also made nanoparticle displayed versions by, we took ferritin-like nanoparticles and attached it through a spike-catcher-spike-tag system. And then when we did all this, we found that with one of our trimerization domains, we got very clean disulfide linked trimers. So you can see here that under, let's see, I don't have, so under reducing conditions, it runs as a monomer, under oxidizing conditions as a trimer.
Can we all switch off our microphones, please, except Professor Raghavan, because we're getting a lot of background noise. Thank you.
Okay, so we made several oligomeric RBDs, and because we didn't want the oligomerization domain to be immunogenic, we minimized that immunogenicity, and we also made nanoparticle displayed versions. So just to summarize all that work, we showed that when we immunized these in mice, there were potent neutralization against even the beta variant, and the sera neutralized all the 4 VOCs in a replicative virus assay, and the trimeric immunogens retained the thermotolerance we had seen for the monomeric immunogens. And so, although we would try to minimize antibodies against the tag, we still got some antibodies. So next and also the PICA expressed protein, even for the trimer, did not give good results with immunogenicity. So then we attempted to design improved monomeric RBD derivatives. And the way we did this is coming back to our yeast display work. So as I said, it's difficult to have screens for improved thermal stability. So the way we got around this problem is we had a saturation library where we had all these mutations. And what we did was we introduced into the background of that saturation library, we introduced a destabilizing mutation. And so now the destabilizing mutation leads to a loss of binding. Okay, so this is the amount of expression, this is the amount of binding. So we get reduced binding. So what we then did was we introduced this mutation in the background of our original library. So we got all pairwise combinations of this destabilizing mutation with all other point mutations in the protein and look for suppressors. And then we were able to get a whole bunch of suppressors which actually improve the binding to the ACE2 target. And then we looked at the thermal stability. We purified the individual proteins and we measured thermal stabilities. And so you can see that there were increases, these are individual point mutants. There were increases in thermal stability in almost all of them. And we combined these together, and then finally we got something that was about 8 or 9 degrees more stable than the original starting protein. And these mutations, the way we did the mutagenesis is we didn't include mutations in the ACE2 binding site. So all the mutations in green, they are far away from the ACE2 binding site. This one is close by, but it points in the opposite direction. And so now so once we had the stabilized mutant, this is just a distribution of the thermal melting temperatures of the single mutant and the double and triple mutants. So you see we've got about 8 degrees of stabilization. And then we looked at the thermal tolerance of the mutants, the wild type and the multi-mutants, using differential scanning fluorimetry. And so this is the wild type protein. So if you expose it at 2 hours at the indicated temperature, above 50 degrees, you can see that the amount of protein, if you cool it back down and do a thermal scan, the amount of the folded protein goes down, right? Whereas now with our stabilized mutant, we could keep it incubated at 50 degrees for 2 hours without any loss at all in the conformation. This was another stabilized mutant. The next one was we kept it at 50 degrees for varying periods of time. So the wild type protein, after 20 hours or so, you can see there's a considerable loss in the amount of folded protein. Whereas with the stabilized mutants, we could keep it for a long time, even at 20 degrees. And this is just 3 stabilizing mutations. So next we looked in adjuvant. So our studies employ publicly available oil in water emulsion called SWE, which has a very good safety record. And so shown here is the adjuvanted formulations. This is the wild type protein. This is the stabilized mutant exposed at 45 degrees for different periods of time. So you can see here that even up to about 1 week to 14 days, we retain a considerable amount of, this is SPR data showing binding to ACE2. So we retain considerable amount of ACE2 binding, even in adjuvanted form at 45 degrees. Of course, lyophilized material is stable for, indefinitely. We have kept it for over a month at 37 degrees and there's no loss in the binding ability. So this is the biophysical characterization, again showing that we have very good binding to ACE2 as well as to another neutralizing antibody. This is the thermal, the increase in stability of wild type versus the mutant. And this is the amount of ACE2 binding that we get after different kinds of storage. So this is storage at -80 degrees, and the P is Pichia express protein, M is mammalian cell express protein. So you can see that even after a month at 37 degrees, so this is the red hash bars and the green hash bars, there's no loss at all in binding to ACE2. The other thing we found out is that the RBD with the beta variant mutations is very rapidly degraded after purification, whereas when we put now our stabilizing mutations, it remains stable at 4 degrees for indefinitely. Okay. So this is a summary of all the biophysical characterization. We get very clean monomeric proteins shown here by secmols, and they are very well behaved. Okay. So next we looked at the immunogenicity data in mice, and really there's what you can do is to focus initially on this. So with the wild type RBD in mice after 2 immunizations, we got fairly weak neutralization titers, so of about 100 or so, whereas with the stabilized mutants, we got several tens of thousands. So you can see here, this is about 30 or 40,000 is the neutralization titer that we got in mice. The Pichia molecules were a bit lower, maybe 3 to 4-fold lower, but still much improved above the wild type. And the Pichia RBD in the wild type background showed essentially no neutralization. The other thing is the neutralization titers. This B1 is the original D614G strain. This is the alpha, beta, and the delta. And you can see that we retain neutralization despite all these mutations. Okay, we did a repeat immunization study, and you can see the results are, if anything, even better than here. We retain high neutralization independent of the variants. For the pikia protein, the neutralization again is a little bit lower, and there is a little bit of a decrease in variants, but still very small. And then we made a cocktail of the beta and the original B1 with the stabilizing mutations. We get very uniform coverage across all the variants. In contrast, if you look at convalescent sera, what you see is that these are a few convalescent, high titer convalescent sera. As Dr. Sanjay Singh says, most of the convalescent sera are down here, a few hundred. And you can see that there are large changes with beta, and there are also changes with the delta variant. Whereas in our case, we are much less sensitive to these mutations. Okay. So this is a summary of the data. We show improved immunogenicity relative to wild type. The mammalian cell expressed protein shows superior immunogenicity to pikia, but pikia is still pretty good. And a cocktail of the wild type and beta shows qualitatively improved immunogenicity. And our sera insensitive to the VOC mutations, whereas convalescent sera are quite sensitive, especially to the beta and the delta mutations. Okay, so now if we want to compare, of course, we don't have human data because it's been a very arduous job to get funded to do clinical trials. But if we look at all the existing formulations and look at their mouse data, and for the same people when they use human convalescent sera, what was the neutralization titers they got? And the important number is in green. This is the ratio, okay? And this is a log scale. So you can see that the very good Moderna and BioNTech vaccines are about 10-fold above the human convalescent sera. They're mouse data. The Chadox is about the same, and ours are really quite high, right? So they're considerably higher. Of course, this is again relative to mouse data. And we need to see eventually when we're able to do human trials what happens. But so far, all the data are quite encouraging. We then did a challenge in transgenic ACE2 mice. This was done at THSTI by Dr. Zegam and Amit Awasthi. And these are the unimmunized mice. These are the unchallenged mice. And these are two groups with monomeric and trimeric stabilized RBDs, which we sent them. You can see that there's essentially no body weight loss in contrast to these mice. If you look at the viral load, this is in the challenged but unimmunized mice. And these are in our challenged but vaccinated mice. So you can see essentially no lung viral load. There's a balanced cytokine expression in these two groups. So this is IFN gamma, IL-4, and IL-17A. So we have a good cytokine response. If you look at the histopathology, this is the unchallenged healthy mice. This is the infected but unvaccinated mice, and these are the mice in our groups. So there is, there's a very, very small amount of lung pathology that's also reflected more quantitatively in these scores. So you can see that these scores are very similar to the uninfected mice, in contrast with the infected but unvaccinated mice. So these are various different scores. So this confirms that the mice are well protected against lung pathology following high-dose viral challenge. Now, this is the data that we have in hamsters. So in hamsters, again, these are the neutralization titers. This is for the mammalian cell expressed RBD, and then this is for the pikia expressed RBD for different VOCs. So the pikia expressed RBD, surprisingly, we took a big hit with the beta variant, and to some extent for the to the Delta variant. With the mammalian cell express protein, that was not the case. However, both of them protected against weight loss. This is the unimmunized, this is the immunized, so mammalian and pichia. And then if you look at all the various scores, the green is the unimmunized and these are the immunized from mammalian and pichia. So we see there's a very good degree of protection from pathology. And these are the actual images. So you can see here that these are healthy, this is infected, unvaccinated, and these are the protected with mammalian and with pichia. So you can see the big difference between this row of slides and this row of slides. So they protect the hamsters against high dose pathogenic challenge. So I'll conclude here. There are a bunch of vaccines and they have efficacies somewhere in this range. Of course, these will all take a big hit with the Omicron, and as they already have to some extent with the Delta. And we've made these immunogens, stabilized immunogens. The mammalian cell express protein shows better immunogenicity than the corresponding pichia express protein. But the pichia is also quite good. And pichia, of course, has the advantage of much cheaper production. So the formulations are highly immunogenic and thermotolerant. They neutralize all the 4 older VOCs. Once we get the Omicron in a pseudovirus in our hands, we can do these studies. And so we think these are promising modalities. And this is where, of course, we are moving forward. And we have ongoing development of antigens for more broadly protective coronavirus vaccines. So I would like to thank all the people who did this work. These are the people from my own lab. So Sami did a lot of the initial antigen design. Dr. Rajmani did all the hamster challenge studies, all the animal studies, the data, the challenge studies. Shahbaz did the yeast display. Devjyoti made some of the pseudoviruses, the mutations for that. And a number of other people have contributed to various aspects of the work. Wahida and Rohan in Dr. Shashank Tripathi's lab contributed the wild-type virus, and Ishika and Somnath did some cryo-EM, which I haven't talked about. The assays with replicative virus initially were done by Shailendra Mani and Shankar in THSTI, and all the ACE2 studies in ACE2 mice were done by Zaigam and Amit. All the pseudoviral assays were done at Dr. Rajesh Ringe's lab in Tech by Sahil, and some of the human convalescent sera is from Dr. Rajesh Pandey and his collaborators at Max. Some of the replicative viruses for the VOCs were all done by Dr. Vasan and his colleagues at CSIRO in Australia. And Minvax, which is a startup I co-founded, did all the animal immunogenicity studies, a lot of the protein purification and serology. These are all the people who have contributed here. These are initial publication in JBC showing the thermotolerant nature of the RBD. This is a more recent publication in ACS infectious disease on the trimer immunogen. And this is one, the most recent one on the stabilized monomer. And the funding, we got very welcome funding from the Gates Foundation, from the Prime Minister's Scientific Advisor's Office, and from Minvax got some amount, a small amount of, well, not a small amount, a few crores from Citibank to help with this work. So I'll stop here now and thank you for your attention.
Thank you, Raghavan, for very interesting data that you shared with us. We can take a couple of questions. I have one question myself. Actually, why do you think that pichia express protein was little less immunogenic than the mammalian cell express?
So we don't, yeah, we don't. sure. We don't fully know the answer. Obviously, it lies in the glycosylation. And so the pichia has high mannose glycans, whereas the mammalian glycans, there are some complex glycans. And one difference was that because pichia hyperglycosylates, there were a couple of designed glycosylation sites in our mammalian antigen that we removed from pichia. We've now reintroduced those back into the pichia protein, and we are awaiting the results of those immunogenicity studies. So we don't have a full answer. It's obviously related to the glycans, but we don't have a comprehensive answer for that very important point.
Thank you. All right.
Dr. Prati, can I just one some yeah. So thank you, Dr. Vardarajan. It's very nicely presented component. My just one, I want to structure biology comments that when you find a mutation, there are so many variants. You know, Anurag has presented sometimes back that you get one mutation and in a protein that is actually inducing kind of I'm not immunologist, so bear with me, a polyclonal kind of response in the body. When one single mutation comes in an epitope, will that mutation will be able to change the vaccine response of all these things, or what is the structural take actually?
Right. So that, yeah, it's a very, very reasonable question now, especially if you look at the beta variant. Okay, so the beta variant, as I showed, has a big drop in neutralization titers for most vaccines, less for ours, but still it was there. And if you look at the beta variant, there are only 3 positions in the RBD which are different from the wild type protein. Now, it's true that the response is polyclonal, but the issue really is that although it's a polyclonal response, there are immunodominant epitopes. And so it turns out that a bulk of the response is directed towards relatively few portions of the protein surface in the polyclonal response. And you know, nobody understands all the factors which give rise to this immunodominance. So, and this has been known for a long time in flu, right? So flu, every year we have to annually update the vaccines, and sometimes the number of mutations is not that large. So it's because of this issue of immunodominance that even though it's a polyclonal response, a few mutations are still able to alter the neutralizing potency. And this is not something that structural biology can readily answer, because you know, we know the structure of the protein, but how the immune system recognizes it and where the antibodies are elicited, how much that varies from one person to the next, one organism to the next, you know, those are very poorly understood things. So.
Yeah, there is another follow-up, just Dr. Prati, I think I'm curious. When we see, this is not in one component, just I want your comment, because if you see the y-axis data of the control versus improved version, whenever we talk about, I think I have seen at several places, the 20 to 30% improvement in the quality of protein or any physiological process as against the background, which a cells always contains, let us say, under in vivo condition, will that make a lot of difference when it comes to the application and function?
Oh, but I'm sorry, it's much more, that amount is much, much larger than 20 to 30%. Let me just, okay, so this is a log scale, right? So these neutralizing titers are 100. These neutralizing titers are like 20 to 30,000.
So I'm not saying for your, I mean, in general, I'm curious, because if you see the control versus the improved version, then the stability, as you have shown, that has shown an improvement of around 25%.
That's right. So the stability, yeah, but the improvement in neutralizing titer with improvement in stability is highly non-linear. That's because, it depends on a lot of factors, again, which we don't fully understand, how quickly the protein might be degraded, how it's presented to the immune system. So, we certainly can't say that always that an increase of a few degrees in stability is going to give you better neutralizing antibodies. But in this case, certainly it was true. That's what the data tells us. But there may be other, especially when you already have the RBD is not that stable of a protein, right? The TM is about 50 degrees. So if you have a protein which already has a TM of 60 or 70 degrees, then further improvements don't really benefit. So even in our case, once we got beyond 7 or 8 degrees and we added more mutations, first of all, the stability didn't increase and there wasn't a dramatic improvement in the immunogenicity.
Thank you, sir. Thank you.
Sure.
Thank you, Professor Varadrajan. Thank you very much for your nice talk. We will move on to the next speaker, Dr. Lalit Kishore, who is COO of the Molecular Biology Reagents and Diagnostics Upscale Program at CChem. Dr. Kishore has worked for many, many years with GE Healthcare and other big companies like BD and Reprocell. He has handled large projects that include the Indian Rice Genome Project, and he has headed the CChem INSDX project under Atmanirbhar Bharat for indigenization of diagnostics in India. Actually, when the epidemic started or pandemic started, the kit had to be imported. Even though kit was assembled in India, all the reagents, most of the reagents had to be imported. And then there was a sea change in the scenario. And I'm sure this is what Dr. Kishore is going to tell us. So Dr. Kishore, over to you. We can't hear you.
I hope you can see my screen and hear me both.
We can see your screen, but go ahead.
Yeah. So, so good afternoon to all of you. And thanks for this opportunity. It's so amazing to hear from the policy perspective, from the research perspective, so deeply from Dr. Agarwal to start with, and then we went on to both Dr. Sanjay Singh as well as Dr. Varadrajan talking in depth about vaccines and vaccine development. But mine is more of a back to the basics kind of a thing. Like you mentioned, sir, at the beginning of the pandemic, at the very beginning of the pandemic, patients were paying close to about 5,000 rupees for an RT-PCR test. And the patients were paying close to about 1,500, 2,000 rupees for a rapid antigen test in this country. The government was purchasing RT-PCR test kits at a price of approximately 2,000 rupees, and was then executing these tests across. Today, we've come to a place where the last tender that closed for the for government's procurement of RT-PCR tests, the governments are procuring RT-PCR tests at 15 rupees per test, 15 rupees. That is the kind of decrease that the cost and the price of these tests has seen in the last 1 year, largely due to multiple indigenous players coming in and both achieving cost reductions as well as achieving they actually managed to bring down the multinational companies' prices down through their efforts. Both of those happened. And one of the small efforts in that direction is the CCamp Index program, which we are working in conjunction, obviously, with DBT, as well as very ably funded by the Rockefeller Foundation. Now, what is the project? What are we doing there? So we have a team of people that is working on different areas where diagnostic companies, indigenous diagnostics companies, need help. We so far have approximately 200 extremely small companies, startups, medium-sized companies, large-scale companies, and even the largest companies of India that are at the forefront of diagnostics manufacture are part of the cohort. Over 500 products, which include end point products like diagnostics kits itself, but we also have ancillary products like RNA extraction, viral transport media, molecular transport media, and then ingredients that go into making all of these things. And that's the reason why when Dr. Sanjay was talking, I said we are down to the ingredient level, and I'll show you a few of the examples of ingredients, but down to all the enzymes, all the chemicals, all of the DNTPs, the amidites, the primers, the probes, buffers, every one of them is actually being manufactured by indigenous companies right now. And COVID came as an unbelievably great opportunities for a lot of these companies to scale up and build world-class companies in their own right. The way that we operate is that we have instituted multiple centers of excellence within the diagnostics environment. Some of the most important aspects where Indian companies need help are quality. They need help with quality and regulatory processes. They need help with clinical testing. They need help in hardcore chemistry, biologics, which comes to protein purification, optimization of processes and things like that. They need help in antigen antibody interactions, because that's one thing where they need help in choosing the right clones and choosing the right antigens, antibodies, etc. They need help in scale up. A lot of these people succeed at the bench level, but when they scale up, like again Dr. Sanjay mentioned, 500 microliters is good, but when we ask them to do 100 liters, 200 liters of that, then scale up help is needed. And then we also have to recognize that informatics is integral to diagnostics. So that's something that we're doing. And last but not the least, a lot of the smaller companies needed great help in business analytics, in trying to estimate where what is needed and kind of connecting them quickly to the need, so that they could produce just in time to meet the marketplace requirements. So those are the 8 centers of excellence that we established at different places across the country. And then to bring all of this effort together, we launched a portal, and this portal was supported by TCS, the Tata Consultancy Services company. They came in and they built in this portal for us. So every single company is registered, both as a user of these technologies and as a customer for most of these products, but also as a seller for most of the products. And then we created this marketplace where people can now go raise wants, raise needs, and their needs are served by each other. And that is what has helped us grow this entire ecosystem. Through our centers of excellence, we've completed close to about 100 projects. The way we take up projects is that we take up any issue that these companies are facing in any of these areas of excellence, and then we actually formulate a solution. We solve the problem for them. We most often create dossiers for them that they can directly take to their regulatory authorities, and a significant amount of this work, thanks to Rockefeller Foundation, has been done free of cost. The and just a few pointers. I mean, while it is very heartening to see the kind of leaps that we're taking in terms of technology, in terms of research, when it comes to some of the basic things like Taq polymerase, like DNTPs, like reverse transcriptase, or just basically making master mixes, we barely, if any, had any companies making this fully in India. And I know that there are a few people who make small quantities of these, but then diagnostic grade is another game altogether. In the last 18 months alone, we have about 20 companies that are making world-class diagnostic grade Taq polymerase. We have about 20 companies making reverse transcriptase. And so as an offshoot, we have multiple people making now RT-PCR ready master mixes in India itself. The most revealing thing was DNTPs. In so many years, we did not have a single company that made DNTPs in India, I mean, which is the core basic ingredient in any of the RT-PCR or PCR based assays. Fortunately for us, in the last 18 months, we've now developed 3 companies that can actually make DNTPs in India. And this is the kind of indigenous contributions that we're making. But getting to the nitty gritty of it, amidites, for example, amidites so key to making oligonucleotides that then are used both to make oligonucleotides as well as probes for RT-PCR. it's not just about bringing down the cost, it's also about ensuring security of supply, it's ensuring speed of supply. The drop in cost has been significant on amidites, and we already have promise from these amidite companies that they'll be able to bring down the cost of amidites even further. These companies, some of them were actually manufacturing bulk intermediary compounds for global companies that were then just doing one-step processes and shipping them back to India at so much of a higher cost. We've actually now managed to bring them into our own gamut, and so that's what's happening with amidites. As a result of bringing down the cost and price of amidites, you can see that the cost, and this cost of the primers and probes is shown as a contribution to the overall RT-PCR test. Within an RT-PCR test, it would cost about 72 rupees for the primers and probes per test. Now that cost has come down to 10 rupees, and we're already seeing cases where it's about 6 or 4 rupees. So, and the number of companies that are actually now making primers and probes has increased from about 1 or 2 companies. Again, might have mentioned that this is significant in terms of diagnostic grade oligonucleotides, that the number of companies has now actually increased to about 6 companies that can make diagnostic grade oligonucleotides, starting from the bench development scale of say about 20 nanomolar scale all the way up to 400 micromolar scale. So we have companies that have the entire range in terms of scale, and most of these came up just in the last 18 months. In terms of master mixes, and this was again something that was completely in the domain of multinational companies, whether it was Thermo Fisher, Takara, Biorad, Kyogen, and these companies were charging us close to about 125, 150 rupees per master mix, per vial. And that cost has come down to 15 rupees. And this presentation is about 2-3 weeks old. Right now, we have a couple of companies that are ready to offer it at 5 rupees per reaction. Again, the number of companies that have actually started making diagnostic grade master mixes for RT-PCR, PCR, and LAMP applications in India has grown to about 5 companies. These are all offshoots that have then helped us now say that we have the capability to produce 100% indigenous kits in India. That means everything. And this is important from the perspective of, we were in a situation where some companies were declaring that they would not be able to send machines to India, they would not be able to supply chemicals to India, that their first priority was either Europe or US. So much for being multinational companies. When push comes to shove, quite a few of them stood back and were reluctant to supply to India at the time that we needed them. And supply times are still, in a lot of cases, extending to beyond 8 to 12 weeks. And in those situations, it is great to have companies that are 100% indigenous companies. But this is not just about RT-PCR. I used RT-PCR as an example, but the same thing has happened with rapid antigen tests, where we can totally become a supplier to the world right now. It is increasingly the case with LAMP kits. So in terms of our capability, if you see in September 2020, we were barely making enough for our own need. Our imports were close to about 80 to 85% when we started off in March, June of last year. But slowly but surely, we've come to a stage where now the government is kind of lifted all export regulations on both RT-PCR and rapid antigen tests. There's a free free export policy, and we are not just meeting the requirement of close to about 1 million tests a day, but in terms of added capacities, we are Indian companies put together just in the RT-PCR domain are able to produce more than about 2 million tests a day. And this is a significant achievement from where we were, both in terms of cost, in terms of turnaround, as well as in terms of absolute capacity. What I'm hoping will happen through this process is that we will have created a manufacturing platform across the country where, and during this process, I must add that we had about 350 or 400 RT-PCR machines in this country, and now we have close to about 28,000 RT-PCR machines in this country. So I'm hoping that RT-PCR based applications, LAMP based applications will only grow, or RT-LAMP based applications will only grow in usage. And with the added advantage of us having worked at bringing down the cost, we will be able to take some of the more sensitive and more specific tests to the wider populations across the country. So, and this is what are we doing now? So, and this is more of a question, or, a kind of a poke, if you will, to all of you to tell us there are variants and there's Omicron and there's more to come. From the patient doctor perspective and from the diagnostics manufacturer's perspective, we know what it is. The doctors tell us that what's the use of bringing out diagnostic kits for Omicron. We still have our own ways of treating, and the diagnostics manufacturers won't exactly go ahead and develop or manufacture these kits unless we show a clear reason why we need to not just detect COVID, but we also need to differentiate. And that is where I think every one of you here will be able to help and tell us how a patient or a doctor can benefit if this differentiation is made. Will Omicron be the dominant variant, or will we still need to check for both Delta and Omicron? What kind of kits must be made? That is some kind of help that we need from the scientific community, so that we can guide the diagnostics manufacturers to develop kits in that direction. I will go from down to up here. I think that from a diagnostics perspective, it's both going to be about surveillance and genomics. So I like to call it surge. But the idea is that we have 100 crore vaccinations. We have such a huge capacity in production of diagnostics. But I think that, and please tell me if we are thinking in the right direction, I think antibody testing is going to be extremely important. Quantitative antibody testing across populations is going to be more important, but that has to go hand in hand with variant analysis and monitoring. And that is where, for example, booster shots, and there is already questions about booster shots. There is questions about novel vaccines coming up. So when do we take booster shots, or what kind of, so will quantitative antibody assays need to be developed so that they will become the basis for, and along with a geographic analysis of where the variants are coming and what variants are coming, will that become a basis for determining who needs booster shots and when? Do kits need to be developed in that direction in a post-immunization diagnostics space? Multiple vaccines, and I'm just saying, with multiple vaccines becoming available, will we need diagnostics that will tell us what my next vaccine should be based on my antibody counts as well as on the geography where I come from? Will Dr. Sanjay Singh at Genova, for example, need a companion diagnostic that says, hey, if you've been vaccinated with vaccine A or B, and if it's already been X months, then this is the time you're ready for a Genova vaccine? Is there a possibility for us to think on those lines and encourage diagnostics kits manufacturers to kind of work in hand with people who are doing vaccines? These are some of the places where we need help. There have been some conversations with people who are talking about prognostic kits. They're looking at both gene expression in host as well as variant analysis to tell doctors what kind of treatment may work or what they may be helping in triaging. And then we're always looking at novel diagnostics, or vaccine recommendation kind of diagnostics. These are the places where we are prodding people within the diagnostic space to currently work on. But I would invite any suggestions, any kind of help that we can get in guiding indigenous diagnostics manufacturers and telling them exactly where they should head as they move forward, in their quest. And be assured that we are a totally different diagnostics country from what we were 18 months ago. We have already established, and Dr. Sanjay and Genova are examples of this, where we've established ourselves as the pharma destination of the world. We've established ourselves as the biopharma destination for the world, vaccine destination for the world. There's no reason why we cannot be the one-stop shop for diagnostics for the entire world, the way we are going. And I'm hoping that while all the tragedy of COVID aside, that specifically in the diagnostic space, we will use this as a huge opportunity to kind of leap into the next era of diagnostics for the country. Thank you. Thanks for the opportunity, Dr. Rajendra Prasad, and thanks to all of you for listening.
Thank you, Dr. Lalit Kishore. We will take some questions. I have a quick query. Are there companies now in India who can make, bulk TACMAN probes, for example?
Absolutely. So we do have enough capacity to actually make 200 micromolar scale, 400 micromolar scale, which is humongous, which could be for a couple of billion tests at one go. We do have people who can make both oligonucleotide simple primers as well as TACMAN probes and BHQ probes at that scale. We absolutely have a few billion tests in one go.
Very good. I mean, I was just looking at, I was Googling for those companies, and unfortunately, again, I only see Merck Sigma.
No. Actually, Merck Sigma is not able to produce at that scale.
I know. I mean, they do only for research purpose.
Absolutely.
But I was unable to find any Indian company. The next company was an American company and a Chinese company on Google. No Indian company showed up. So do something about it so that we become aware of Indian companies able to provide this.
Absolutely, sir. Absolutely.
Dr. Varti, can I have a question?
Yeah, please go ahead.
Yeah. So, Dr. Lalit, I found it quite an interesting presentation. Unfortunately, I was disconnected in between due to internet issue. Sorry for that. So, I am having a question regarding your quantitation of antibodies which you are talking about. So, still, when we talk about antibodies, we have a group which talks about overall antibodies, another group just neutralizing antibodies. And still, we are much focusing on neutralizing antibody titer when we talk in terms of protection, but we don't have any idea what is the level of, what is the threshold, means what should be the cutoff from where we can say this is really the level of protection can be demonstrated from here or not. So just I would like to have your comments on that.
So I completely agree with you that that's the debate. And we do have developers who have developed specific ELISA-based assays for the overall antibody counts, but they have reasonably well-established quantitative techniques. These are ELISA. Our focus has been more on how can we make this more accessible to people, because unfortunately ELISA machines are not everywhere. So we've had developers who have come, for example, with dry blood spot kind of paper kind of collection that can be done, and then they've done concordant studies with established ELISA kits. And now, they come to us and they say that, look, we can actually do remote collection and centralized analysis. But we are looking for people who are coming up with more specific assays that can not just give you the overall antibody, but can give you some kind of a neutralizing antibody assay, but something that can be accessible to larger populations so that it serves the purpose of larger serosurveillance and vaccine recommendation. And then I think that effort has to go along with the genomic surveillance that's happening, and that's where this thing needs to come together. But that's the future for diagnostic companies now.
Thank you.
Yeah, Raghavan, you have some question?
Yeah, I have a question and a comment about the ELISA titers. So they scale very well with neutralizing titers. So I think it's perfectly reasonable to just measure the ELISA titers. I don't know that the ACE2 competition assay that whether it's worth the additional effort. Because they're neutralizing antibodies outside the ACE2 site also. That's one just comment. The question I had is, or the requirement is, all our gene synthesis we need to get done from outside, and there's a real need, because it takes forever It doesn't take forever, but it takes a few weeks. And so right now when the Omicron has come, everybody abroad already has pseudovirus, they have everything. And here we are just sitting, twiddling our thumbs because it will take GenScript 3 weeks to give us the material. And so this is a real need. There are plenty of oligos,
Yes.
but as far as I know, or at least we have not aware of people who will do gene synthesis for us.
I heard from my own sources that I know that Dr. Sanjay Singh is not saying anything, but Genova has the capability to do it, but I guess they're using it for their internal research programs right now. And I know that Syngene in the Biocon company also has the capability, but again, very, very busy in internal research programs. But point taken, and there is instrumentation available from Codex that can actually help you synthesize, say, 32 constructs in 24 hours. And there's no reason why we should wait for GenScript.
No, but Codex has to send you the cassette.
Yes.
So it comes to the same thing.
True.
So, we can do gene synthesis by getting oligos and so on. But again, that's a waste of our time. Right? There should be somebody here who can just do it and give it to us.
Absolutely. So point taken, definitely part of our proposal going forward. I mean, I'll keep you updated, but absolutely on target there.
Sure. Thank you.
Yeah.
So Dr. Raghavan, we have the Codex facility available in-house, and still our turnaround time is 2 weeks because plate has to come and just we do it a very high speed rate because the kind of contractual agreement we have with them. But now I think they are coming and they are establishing this in India, but it's still maybe 6 to 8 months back because we want our gene synthesis to be done in a week time.
Yeah.
And they are very worried about their intellectual property and all that. So we have signed the agreement, but let's see. I want that turnaround to be 1 week because for us making Omicron was only taking 54 days because 21 days will go for the gene synthesis.
Yeah, exactly.
All right. Thank you very much. I think we have already overshot the time, so we will stop here. I would like to thank all the 4 speakers for their excellent presentations and very timely subject that was chosen by the organizers. So thank you very much. It's over to the organizers, Dr. Pathak now.
Thank you, sir. So this session of COVID-19 and Indian perspective was very wonderful, interactive, informative, and interesting sessions. We learned actually recent update of COVID-19, trends of pathogenesis, vaccine development, and diagnostic as well. So this is a really good lesson for us. So I would like to place my heartfelt thanks to all the speakers, Dr. Anurag Agrawal, Dr. Sanjay Singh, Dr. Varadarajan, Dr. Lalit Kishore, and Chair Professor Sudhanshu Bhrati, and eminent scientist of India for sparing very precious time to attending the session despite a very busy schedule. So thank you so much, sir. Thank you.
Pathak, before we go, I think Mac wants to take a picture of everybody. So leave your camera on for a second, please. One of you. Meantime, I'm really very thankful to all of you, all the speakers, wonderful talks, very controversial session, and I'm very pleased that it was very interactive. And we have learned a lot, and I feel very safe that at least you guys are thinking about us. So it's a wonderful feeling.
Thank you, sir. Done.
Okay. So we can log off now. Yeah, thank you very much.
Bye.
Right. Bye-bye.